Thank you, Stan, for the introduction. Good morning, everyone. So I'm going to tell you about our work on developing more effective treatment for persistent Lyme disease. But first, I'd like to thank Olivia, Holiday, and Stan, and the LivLyme Foundation for the kind invitation. It's great to be here. So let's see. Okay, here's the outline of my presentation. So I'm going to start with a brief overview of the complexity of the disease and then tell you about the bacteria that's really quite intriguing, that can develop different forms, and then tell you about the persistence problem in Lyme disease, including PTLDS, and then show you that Borrelia can develop persister bacteria, and then show you the limitation of the current Lyme antibiotics for treating such disease conditions. And then I'm going to show you that TB treatment could serve as a good model for developing more effective treatment for persistent Lyme disease. And then I'm going to show you that we have went on to identify drugs that are effective at killing Borrelia persisters in vitro. And then we went on to do the animal testing in the mouse model and show you that this approach actually works. We were able to identify drug combinations that are not only active in vitro, but also active in the mouse model of persistent Lyme disease. So then I'm also going to show you our work on essential oils. And some essential oils turn out to be very effective in vitro, and also some natural products that are being used by some physicians, and we found some of them to be quite active as well. And then I'm going to show you our new model for PTLDS due to persister inocula, which is quite interesting. And then I'm going to tell you about two types of persistence mouse models. One is the one we recently developed, start with the persister inocula, and the other type is a long-term persistence model that takes about 2 years to evaluate the kind of drug combinations and show you some data that indeed we're able to find effective drug combinations that eradicates persistent Borrelia bacteria. And then I'm going to finally discuss the strategies for treating more effectively persistent infection of Borrelia, and end with some future directions. As Zhang alluded to, in fact, Lyme disease is a pretty complex disease. It has different stages, starting from early localized, early disseminated, to late disseminated stage, and then PTLDS. If you look at the treatment, but it's quite simple. It's a monotherapy with doxycycline or amoxicillin or cefuroxime for only 2 to 4 weeks. This kind of treatment for the early stage can be effective for about 80 to 90% of the patients, yet still 10 to 20% of the patients do not feel better. That's a PTLDS, right? As a disease stage moves on, it's a very heterogeneous disease, then the success of treatment can become increasing less until the PTLDS stage. The current treatment does not work anymore, or does not work very effectively. Sorry, I went too far. Yeah. So we can say that everything surrounding Lyme disease about the controversy is due to this PTLDS, post-treatment Lyme disease syndrome. Dr. Alcock said it partially treated Lyme disease syndrome. It's a problem. Everything we can say, all the controversy is surrounding the nature of the PTLDS. Why these 10 to 20% of patients do not get better after standard treatment? In fact, if you look at this problem, if you look at the infectious disease treatment in general, in fact, you find that this may not be unique to Lyme disease, in fact, because there are other situations like PANS, PANDAS, even reactive arthritis. that can have similar sort of a situation where if you treat acute phase of the disease, it's relatively easy to treat, but it's the persistent chronic form of the disease. The current medicine does not know how to deal with it. It's very likely it's due to form of a persistent infection or biofilm infection where a single antibiotic is not really effective in getting rid of such condition. So these patients suffer from, I think John alluded to, this persistent fatigue, joint muscle pains, and also brain fog. And the number of such cases is estimated to be close to 2 million by 2020, which is next year, which is quite amazing. So then if you look at the various clinical trials for treating such conditions, they don't usually work very well. Though 2 actually reported no effect at all, the Klempner trial here and also the Dutch trial. And 2 studies report some effect of improved symptoms, but it's not a cure, okay? Okay, so there is no FDA-approved treatment for this condition. Patients take all sorts of medications, and they don't work very well. So it's a huge unmet clinical need. The key question is, what causes this PTLDS? There have been different theories for this. John, I think, alluded to that already. First one is due to host response to dead bacterial antigenic debris. The second is autoimmune. The third is co-infections. And the last one is persistent infection. Due to the current antibiotics is not very effective at killing such persistent form of the bacteria. So in a patient, in fact, all these 4 possibilities can potentially coexist. And I think it's possible to address this question. My personal view for this is that the common driver for all these 4 different possibilities is a persistent infection. And we can test if we have a more effective drug combination regimen that eradicates the persistent bacteria, then we'll see what happens to the other conditions, whether they're still there or they're gone as well. So in support of the persistent theory, there have been different animal studies in mouse, dogs, and monkeys that show that after antibiotic treatment with a single antibiotic, you typically do not get the organism cultured, but by PCR, you can get a positive DNA test. That means the bacteria somehow may still be there. I think the most convincing study of a persistent infection was demonstrated by Steve Barthold and Amir Hotzik in 2014, when they showed that after the antibiotic treatment, if you wait, there was no DNA detected by PCR, but if you wait for a year, then you get a sudden resurgence of bacterial DNA. So the only explanation for that is the bacteria replicated, but in a form that cannot be cultured, which is very intriguing. So this resurgence phenomenon is very interesting, and we are able to develop drug combinations that can eradicate the resurgence. I'm going to show you that in a little bit. So then there have been also anecdotal human studies, not really systematically, that reports in patients after treatment. Sometimes you can still identify, culture the organism, sometimes it's PCR, but it's not consistently. That's why it's easy to deny it's a persistent infection. So this is unlike tuberculosis or staph infection, when the infection returns and you can culture the organism. But for Borrelia, you cannot, often cannot. This is the biggest problem facing the field. There's a problem with the culture methodology that is not quite there. And of course, if one can develop a better treatment that eradicates all this, then patient symptoms can go away. Then there will be proof. So we are actually working on that. So the bacteria is very intriguing. So I think previously people's attention have focused on this spirochete form, which is the growing form of the bacteria. But then the bacteria under stress condition in old cultures developed these variant forms, which are persisters, the aggregated form, which are more tolerant to antibiotics. The current Lyme antibiotics do not kill them. these are actually persistor forms, the variant forms. So we're able to show that, in fact, in vitro, we and others have shown Borrelia-developed persisters that are not killed by the current Lyme antibiotic. And then you might wonder why it's important, why the persisters not killed by the current antibiotic important. So we have this cartoon, many of you probably have seen this, which is actually shows the importance of the persisters and also the drugs targeting persisters. So the mower is a current Lyme antibiotic. It removed the top part, you still have the root here, and after removal of the antibiotic, you get the relapse of the disease. And what you need is this shovel, persistor drugs that dig out the roots, the whole thing is gone. Then you need really a combination of the mower and the shovel in order to more effectively cure persistent infections. So it turns out in tuberculosis, we have this treatment model paradigm. That's exactly this. That is, you need to include drugs that kill the growing form of the bacteria, like isoniazid. Then you need drugs to kill the growing and non-growing population as well, like rifampin. Then you have parazenamide that exclusively kills the non-growing form persisters. So you need all three drugs in combination in order to more effectively cure the persistent TB infection. So TB people have accumulated nearly 70 years of experience for treating tuberculosis. This can be borrowed for developing more effective treatment of persistent Lyme disease, which is, we don't have to spend 60 years to develop more effective treatment for persistent Lyme. So what TB people have learned is that you need a drug combination to prevent drug resistance, okay? 2 drugs to prevent drug resistance. The other is a drug combination is also important for increasing the efficacy of the therapy. As shown here, single drug isoniazid, you have a 37% cure with a 3-month treatment, whereas if you add streptomycin plus isoniazid, cure is increased to 67%. TB people also learned you need to really treat sufficient time. That's why in the 1950s, treating TB is 2 years with streptomycin, PES, isoniazid. And if you treat shorter for 6 months, you get a high relapse rate of 62%. If you treat for a year, still high relapse rate of near 20%. You need to treat 2 years, then the relapse is low, is 4%. That's why in the 50s, treating TB requires 2 years. Okay, then TB people also learned the importance of persistor drugs. Then adding one persistor drug, rifampin, shortened the therapy from 24 months to 12 months. Then adding parazenamide, shortened the therapy to 6 months. That's why we need to apply the same sort of principle. So in the TB field, we know the importance of persistor drugs. As a persistor drug, parazenamide, another persistor drug is clofazimine. Okay, which is amazing. These 2 drugs turn out to have activity against Borrelia persistor as well in the combination setting. So I'm going to show you the data. So we're trying to apply the PZD TB treatment paradigm to developing more effective treatment of persistent Lyme disease. So in the beginning, we didn't have drugs active against Borrelia persisters. We have to develop a method called CyberGreen PI assay, and then use this rapid screening method, screen FDA-approved drug library, etc., and identified a range of drug candidates that are quite active, including daptomycin, clofazimine, sulfur drugs, artemisinin, ciprofloxacin, anthracycline drugs, anti-cancer drugs. So this is actually the assay we use, the CyberGreen PI assay, where it stains the live cells green, dead cells red. If you add a drug, if the drug is effective, you see fewer green cells, more red cells. Based on this change of red-green cells, we can very quickly determine if the drug is effective. So using this assay, we were able to, in fact, rank the current Lyme antibiotics in their relative activity in killing Borrelia persisters. This actually turned out to correlate very well with clinical observation because by far the most effective current Lyme antibiotic is the cephalosporins, like ceftriaxone here. Is the lower the percentage, the more effective the drug activity. So these doxycycline, amoxicillin, not very effective, 60-70% range. Interestingly, tetracycline is turned out to be more active in the assay that correlates with the clinical observation, seems to be more active than doxycycline. And then importantly, we find persistent drugs, daptomycin and the clofazimine, etc., more active than the current Lyme antibiotics. These persistent drugs act differently from common antibiotics because they have different targets. Often, it's the membrane, cell membrane is a critical target for persistent drugs. And also, DNA. These persistent drugs can damage DNA and also produce reactive oxygen species. So these are very different way the persistent drugs work. For example, daptomycin, clofazimine, artemisinin essential oils, these are not likely to act on the membrane and kill the persistent bacteria. And then we have drugs that can damage DNA like the anthracycline, donomycin, very potent, very effective at killing persistent, but of course it has side effect. But then clofazimine, those are nitramidazole drugs, and the cephalosporin can produce reactive oxygen species and damage DNA that way. So these actually are different from the common antibiotics, which actually inhibit the cell wall synthesis, protein synthesis, nucleic acid synthesis. These persistent drugs act differently. So we showed that, in fact, even we have persistent drugs, we cannot use them alone. We have to use them in the context of drug combination. And we show that, in fact, it's a triple drug combination. This was already published, so I'm going to be very quick. So we found daptomycin, doxycycline, cefuroxime. 3 in combination can completely eradicate the aggregated form of biofilm-like structures of Borrelia, whereas single antibiotic or 2 drug combinations are unable to do so. So the issue with daptomycin drug combination is that it's IV antibiotics, very expensive. So we're interested in developing oral regimens that are more affordable. So this actually, we are involved in doing hundreds, thousands of different drug combinations because there are different possibilities. So how do we test them, right? So we have a guiding principle of using this yin-yang model persistent drugs. We know we have to include a persistent drug. Then other drugs that kill different bacterial populations in the context of yin-yang model. Then the idea is to, through hundreds of these drug candidates, we're able to develop a bunch of these 3 drug combinations and then evaluate them in the mouse model. In the meantime, we also found some essential oils to be very active, very interesting, because we saw online that some patients use these essential oils, and we don't quite know. So we actually decided to test some of them, and it turned out to be extremely active. For example, we had done 2 studies by now. In the first study, we showed oregano and the cinnamon bark and the clove bud to be very effective, completely eradicate all the Borrelia persistent even at 0.12%, which is a pretty low at the concentration, but even seem to be more active or similar to the most active persistent drug, daptomycin, we found. So in the second study, we found another 5 to be very effective, including garlic, allspice, myrrh, hadicam, lycicubeba. Interesting, we also found that essential oils in combination can enhance the activity of antibiotics. Here, for example, we found that oregano and the cinnamon bark can enhance the activity of doxycycline plus cefuroxime. These are 2 currently used Lyme antibiotics. So in subculture studies, as you can see, oregano plus the 2 current Lyme antibiotic drugs completely eradicate all the bacteria. Other essential oils do not seem to be effective. As you can see, still some live bacteria remaining in the combination. Interestingly, we tested this fibroblast cell model called ex vivo fibroblast cell model. We use this GFP, Borrelia, infect fibroblast, then treat with different antibiotics. Interestingly, the current Lyme antibiotics actually does not really have much of an effect. Somehow the cells protect the Borrelia bacteria from being killed by antibiotics. that provides some kind of a persistence. Then if you can see here, if you add daptomycin is more active than the current Lyme antibiotics, doxycycline, cefirozim, then the triple antibiotic can eradicate in the absence of the cells. But in presence of the cells, some Borrelia are still alive, are protected. Then, in combination with the essential oil, oregano or cavacrol, the active component of oregano, can completely eradicate these, even the cell-associated Borrelia persisters, which is quite interesting. So we're interested to move on to evaluate these in the mouse model. Yeah, I'm going to skip this. So this is the cavacrol. So which is the active component of oregano, and which actually we are able to show that it's possible to develop this into in vivo testing. So I'm going to skip this for now in the interest of time. Then we also evaluate a range of herbal medicine, natural products in combination. This is the work in collaboration with Sanjay Shraig and Jacob Leon. Interestingly, we evaluated these were used in the Buhner book, and turned out some of them turned out to be very active against Borrelia. For example, cryptolepis, Japanese knotweed, cat's claw, black walnut, artemisia. Okay, they turn out to be very active against the Borrelia, both the growing form and the non-growing form. In fact, we found andrographis, which was claimed to be having antimicrobial effect, did not work, did not have activity. So is stevia, did not show activity in our assay. Group seed extract, silver, monolaurin, etc. didn't have effect. So we're interested to move on to test these in vivo, perhaps. And then moving on to the next part of the in vivo testing. So we have this two types of persistence model. The first one is the long term. It takes about 2 years. And you can only do a few such experiments, then you have to retire. So that's why we cannot wait. And we get asked, when can we get in vivo data, right? So in the meantime, it actually forces us to think hard and try to develop more rapid persistent model, which we're able to, I think, get at least one way to create a more rapid persistent model, that is to use its different forms. We found if you use different inoculum, the log phase, the microcolony, the biofilm-like structure, and also stationary phase, these three different inocula used for infecting mice, it turns out that the microcolony form and stationary phase form create a more severe disease, a more persistent disease that's not easily cured by the current Lyme antibiotic. So using this model, so this is the mouse model that shows you the joint swelling that's more obvious with the microcolony inocula, and somewhat less so with the stationary phase, and even less so with the log phase inocula. But by later time point of 35 days, the difference among the groups becomes less because the log phase actually swelling can catch up. And this is actually by histopathology studies that's also consistent with more severe inflammation in the joints and in the heart base. I'm going to skip that. And then based on this, we came up with this new model for PTLDS. That is, whether or not the patient is going to get PTLDS is determined from the very beginning of the tick bite. So if some patients may be unlucky, okay, they just inoculated with a very severe form of biofilm structure, so that even though you treat with a single antibiotic, you're not going to cure it. Other patients are more lucky. They inoculated with some form of a bacteria, maybe more primary spirochete form of the bacteria, that can be treated, that can be cured if you start the treatment early enough, okay? So we actually propose this type of divide the persistent Lyme disease into two types. One is this type 1 persistent Lyme disease, which is essentially PTLDS, okay? The other is this long-term persistent model. That is, the patient are not diagnosed or treated in time. And then after some months or years, then patient found to have persistent Lyme disease. This is the form that's called type 2 persistent Lyme disease, and it's probably more difficult to treat. So we show here the treatment results for using this more rapid persistent infection mouse model, where we created these two types of infections. One is with the log phase bacteria inocula, the other with microcolony, the biofilm persister inocula. As you can see that the current Lyme antibiotic cannot, like doxycycline, is unable to clear out in either animal models. Ceftriaxone is pretty active, is able to eradicate in this log phase inocula mouse model, but not so much in the persister inocula. Other drug combinations, doxy plus ceftriaxone, not able to do so. Our triple drug persister drug combination is able to completely eradicate with the daptomycin, ceftriaxone, doxycycline. Even in the persistent model here, by culture, it's completely eradicated, and also by PCR, this is all gone. So then we went on to do this study of this long-term persistence mouse model. This study was done in collaboration with Monica Ambers, where the mice infected, then treated for 2 months, actually left for infection to establish for 2 months, then treat for 1 month, okay? Then at different time points, 6 weeks after treatment, and also 12 months, 11, 12 months after treatment, we do 2 xenodiagnosis for the tick to pick up any residual bacteria, a sign of a persistent infection, as well as at the end of the experiment, we look at the mouse tissues by culture, by PCR. And this is part of the results, it's just highlighted here. It turned out we found some drug combinations to be highly effective, like clofazimine plus doxycycline plus cefuroxime can completely eradicate Borrelia by culture and also by PCR, both in the ticks as well as in the mouse tissues. Gone. The DNA is completely gone. So this is the more extreme form. So when you cannot culture, the DNA is still positive, you can say the bacteria may be dead, okay, may still be there, may be dead. But then when the DNA is gone, that's more stringent proof that the bacteria is indeed eradicated. So we show that not only for the clofazimine drug combination, we found that for pyrazinamide plus doxycycline plus cefuroxime as well. So this is a potentially very exciting results. I think this is probably the way that can lead to the clinical studies, we hope. So what's for the future of treatment of persistent Lyme disease? So I think in the short term, we still have to use this two-tiered approach, the IDSA 2 to 4 week antibiotic. For those unlucky ones, these are the candidates for testing the persistent drug regimens. But in the long term, if the persistent drug regimen indeed works and cure such PTLDS patients, then we can start perhaps treating these Lyme patients fresh, okay, using the persistent drug regimen, so that 10 to 20% PTLDS patients do not even happen. So that's the goal, okay? So for the future then, I think it looks very promising at this stage, because we're able to identify persistent drug combinations that the current Lyme antibiotics are not able to achieve. Okay, so in the meantime, we're in the process of developing more effective drug combinations in vitro in the mouse model. And I think in the meantime, based on the promising in vivo mouse data, we can start clinical trials to test these regimens on PTLDS patients, as well as on antibiotic refractory Lyme arthritis patients. And then we need to look for biomarkers for treatment response. For example, Zhang talked about this brain imaging. We want to see whether treatment with persistent drug regimen, the brain inflammation can be eliminated, right? We want to address the role of a co-infection and immune suppression. And of course, we need support in order to do this. Finally, I want to thank people who've done the work, and also our collaborators, Monica Ambers, Emir Hodzik, Zhang Alcott here, and some other physicians. And in particular, I want to thank the founding agencies, Cohen Foundation and DOD supported our drug combination studies. Global Lyme Alliance and LivLyme Foundation supported the essential oil studies. And also Bay Area Lyme Foundation supported the natural products herbal medicine studies. I also thank NetCap Lyme and Steve Sim for their support. I'm going to stop here and be happy to take any questions. Thank you.